Hi, my name is Mike Kohanski, and this is a chapter on sinonasal physiology and function. I have nothing to disclose. So in this chapter, we'll start with an introduction, followed by a discussion on airflow in the nose, briefly touch on olfaction, and then discuss immune defense, specifically focusing on mucus and goblet cells, cilia, mucociliary clearance, followed by wrap up with some conclusions and key points, suggested reading, and sources. So why do we have sinuses? What are the function of the sinuses? So the sinuses have a number of functions. They can contribute to voice and vocal resonance. They have a role with humidification and warming of inspired air. They can increase the olfactory surface area. They help to absorb shock to the face and head from a blunt trauma. They provide thermal insulation for the brain, contribute to facial growth and lighten the skull and facial bones, and serve as an initial immunologic point of contact for inhaled or respired particles, allergens, and pathogens. So now we'll discuss airflow in the nose. So nasal airflow allows for a number of things to happen, including in the nose. So filtration of particles, as well as humidification and warming of inspired air. And nasal airflow is going to be regulated in part by the cross-sectional area, which will have both inflammatory and anatomic contributions. And some of the anatomic locations that would be important for regulating airflow would be the region of the nasal valves, the length of the septum, the inferior turbinates, as well as the piriform aperture. And focusing in on the inferior turbinates, the size of the inferior turbinates relative and how much they can fill the nasal passages may regulate turbulent airflow in the nasal valve region. So when thinking about the regulation of the inferior turbinates and how their size fluctuates, that happens through autonomic regulation. And so specifically parasympathetic stimulation of the inferior turbinates will lead to increased venous vessel dilation and blood flow, so an increased capacitance of the inferior turbinates, which will lead to an increase in the size of the inferior turbinates, which would result in an increase in nasal obstruction. And at the same time, parasympathetic stimulation increases secretion from goblet cells and submucous glands, which would result in rhinorrhea. So in sum, parasympathetic stimulation of the inferior turbinates leads to an increase in obstruction and rhinorrhea. And on the flip side, medications like oxymetallazine or phenylephrine are sympathomimetic. So topical application intranasally of these medications would lead to constriction of blood vessels. This would reduce venous capacitance and shrink down the size of the inferior turbinates. This would lead to a reduction in intranasal resistance and an increase in airflow. So is there a relationship, and what is the relationship between airflow in the nose and sensation of nasal obstruction? And part of that question is, where is the majority of the airflow in the sinonasal passages? And the majority of the airflow is likely through the nasal passage, and where the amount of airflow is going to be regulated in part by the nasal cycle, which is the normal asymmetric swelling and shrinking of the inferior turbinates, where one side will swell up while the contralateral side will shrink down. So the side that's swollen will have decreased airflow and increased obstruction, and the side that is where the inferior turbinate is shrunk down will have increased airflow on that side. And that will vary from side to side throughout the day, and that's part of a normal cycle. And a question is, where exactly is the greatest area of obstruction occurring? And from some computational fluid dynamics studies at normal resting breathing, about 50% of the pressure drop occurs at the nasal valves, again pointing to that area as an important location for regulation of airflow and perhaps for where nasal obstruction may occur. What was also interesting from some of these studies looking at normal resting breathing is that there may be greater airflow through the middle meatus than through the inferior meatus, suggesting a role for the middle meatus and the middle turbinates in the regulation of airflow and the sensation of nasal obstruction as well. So now we'll move on to discuss olfaction briefly. So olfaction in the nose, there are two nerves that can sense odorants in the nose. There's the olfactory nerve, cranial nerve 1, and the trigeminal nerve, cranial nerve 5. And the olfactory nerve, cranial nerve 1, connects to the olfactory epithelium and the olfactory cleft, and has extension, that olfactory epithelium has extensions along the posterior septum, as well as the superior and middle turbinates. And at physiologic airflow rates, about 15% of inspired air flows through the olfactory region. And odorants are carried to the olfactory cleft by nasal airflow, and odorants absorb and diffuse into the mucus and are carried by odorant binding proteins to olfactory receptors, where they can then stimulate them. And olfaction has a significant contribution to our ability to taste things. And so to separate that from the taste element that we have on our taste buds, which is gustation, which is a direct interaction of those taste molecules with the taste buds, and those are specifically sweet, salty, bitter, sour, and umami. And the contribution of olfaction to that is that retronasal airflow can stimulate olfactory receptors, and that gives us more of those complex flavors that we get with taste. And that's how olfaction in part can contribute to our taste sensation. And changes in olfaction are multifactorial and can include a number of different reasons, but one of those can potentially be just a failure of the odorants to reach the olfactory cleft. So if an odorant can't get there, then a person's not going to be able to smell or detect that odorant. So, and that can occur through disruption to airflow that would reduce the amount of odorants that can make it to the olfactory cleft, or perhaps something that might disrupt or decrease the ability of odorants to move through the mucus, among other reasons as well. So now we'll move on to discuss immune defense, and specifically focus in to start on mucus and goblet cells. So to start this discussion, start with just a definition of what we're talking about, what we're looking at. So the first thing is to define the airway surface liquid. And as you see in this, this picture here, the airway surface liquid is comprised of a periciliary liquid layer, a PCL layer, which is a very small layer of liquid that is overlying the epithelium. And above that periciliary layer is a mucus layer that sits on top of the PCL. And the mucus is rich in mucins, and a number of the mucins are secreted by goblet cells. So the airway surface liquid is important, and the depth and viscosity of the airway surface liquid can help determine how fast that liquid can move across the epithelial surface, and among other things. And that depth and viscosity of the ASL are determined in part by ion concentration. So with a healthy, intact epithelium, with an intact barrier, active transport of ions across the apical surface is going to regulate the depth and the viscosity of the ASL. So in particular, it's going to be governed by movement of chloride and sodium ions across that apical surface. And as you pull more sodium chloride ions onto that apical surface by osmosis, you're going to move water to that surface as well, which is going to affect the ASL. So for example, if hypertonic saline is applied to an epithelial surface, that would reduce the mucus viscosity because there would be an increase in osmosis of water into the ASL due to the increased amount of salt molecules. And another example of this is in cystic fibrosis, where CFTR mutations, which is the cystic fibrosis transmembrane conductance regulator that regulates chloride transport, which has various mutations in cystic fibrosis, and that chloride transport is disrupted, and that leads to dehydration of the ASL and mucus stasis. So the ASL has a number of functions. So the mucus has a barrier function where it prevents loss of water from the epithelial surface, and also is sticky and can filter inhaled particles and substances. And there's an important immunologic function as well to the ASL and the mucus, where it contains antimicrobial peptides, lysozyme, lactoferrin, secretory IgA, elements that can serve as a first line of defense to a respiratory pathogen. So mucus itself is a gel layer that consists of water, ions, proteins, and other molecules. It coats the respiratory epithelium, which you can see depicted here in this picture, showing a picture of a left middle turbinate. And we make about 0.8 to 1.6 liters a day of mucus. And mucus is comprised, again, there are mucins, which are part of mucus, and mucins are produced by goblet cells, which you can see here. There's a cross-section of a goblet cell as seen on an electron microscopy image. You can see the goblet cell here, which has these secretory granules that are mucin-rich. And mucins are glycoproteins, and they can be secreted, they can be gel-forming, they can also be cell surface associated. And mucins themselves also have antimicrobial, anti-protease, and antioxidant activities. And again, they can be sticky, and they can capture bacteria and viruses as well. And the major mucin that's produced by goblet cells is MUC5AC, whereas MUC5B is typically produced more by the submucosal glands. So the key regulatory proteins that govern goblet cell and goblet cell differentiation, there's a SAM-pointed domain containing ETS-like factor, SPDEF, which regulates the goblet cell transcriptional program by Forkhead box A3, FOXA3. And SPDF overexpression is known to induce goblet cell hyperplasia. And goblet cell hyperplasia has been observed in the airways of patients with asthma, COPD, as well as chronic rhinosinusitis. And that hyperplasia can be driven by type 2 inflammatory mediators such as interleukin-13. So now we'll move on and talk about cilia. So cilia are hair-like organelles on eukaryotic cells, and the structure of the cilia is important. So they're about 6 microns in length. They are about 0.2 to 0.3 microns in diameter. And they have this axoneme structure, this 9+2 microtubule structures you see depicted here, where there's paired microtubules, where you have the paired central structure surrounded by these 9 doublets. And then there are these little dining arms that are attached to the microtubule structure. And the movement of these dining arms helps regulate the movement of cilia, and that occurs through an ATP-dependent fashion. So ciliated airway epithelial cells are multiciliated, as you can see in this electron micrograph picture here, showing ciliated airway epithelial cells. And there are about 200 to 300 cilia per cell on these airway epithelial cells. And the major transcription factor associated with ciliogenesis is Forkhead box J1, FOXJ1, which this transcription factor process helps promote basal body docking to the apical membrane, which then supports axoneme formation and subsequent cilia formation. So ciliary beating is directional with a fast and a slow phase, and ciliary beat frequency in the upper airway is about 10 to 15 hertz. And this is just a short video here showing that ciliary beating. And you can kind of see that the flickering of the cilia is occurring mostly in a single direction here. And there are a number of conditions, compounds that can both increase or decrease CBF, ciliary beat frequency, and some compounds or situations that can increase CBF include application of acetylcholine, an increase in temperature, as well as application or release of ATP. And so these processes are calcium dependent and regulated in part by cyclic AMP, cyclic GMP, and the associated kinases, protein kinase A and protein kinase G. So ciliary beat frequency can be decreased by virulence factors secreted by pathogenic bacteria and fungi, including aflatoxins secreted by Aspergillus, pneumolysins secreted by, released by Streptococcus pneumonia. And decreasing temperature will decrease CBF as well, and topical application of dexamethasone or fluticasone can also lead to a decrease in ciliary beat frequency. And not only do some pathogens decrease CBF, but in some cases, there can be a loss or a shedding of cilia as well. So some viral infections like rhinovirus and influenza can lead to shedding of ciliated epithelial cells. And cilia also can have function as sensors, and airway epithelial cells also can have a sensory function. So one of these functions is chemotransduction with bitter taste receptors. So one of the bitter taste receptors, T2R38, has been found in cilia of airway epithelial cells. And stimulation of this bitter taste receptor can result in an increase in ciliary beat frequency and nitric oxide production. And airway epithelial cell cilia can also have a mechanotransduction properties as well, where they're able to sense shear force and osmotic shifts through these TRPV channels, these transient receptor potential vanilloid, which are calcium channels. And the way that they respond to these changes in mechanical forces can be through changes in mucus secretion, ion channel function, as well as ciliary beating. So now we'll put together this component of mucus, goblet cells, and cilia, and talk about mucociliary clearance. So mucociliary clearance consists of mucus production and movement of mucus by ciliary beating. And as you can see here in this picture, you have the directional movement of the cilia, which is going to promote the directional mucus of the ASL components, or the PCL and the mucus. The mucus will serve as a filtration system to help capture particles, microbes, fungi, viruses, and then help move these different trapped particles and molecules out of the sinuses and to be cleared. And this serves as a primary innate defense mechanism to help limit and reduce the buildup of potentially pathogenic or harmful substances in the sinuses. And the way the mucus flows out of the sinuses is going to be toward the nasopharynx and the oropharynx. So for the the frontal and the ethmoid sinuses, the direction of the mucus flow is also going to be in some ways assisted by gravity, and it's whereas in the maxillary sinus, the direction of mucus flow is up toward the drainage from the natural os out into the middle meatus. And when the mucus drains from the sinuses, it will then drain toward the nasopharynx and the oropharynx, where the mucus is then either swallowed or expectorated. So with the maxillary sinus, where the direction of mucus flows toward the natural os, so against gravity, if there can be a situation where the mucus can recirculate into the sinus, called maxillary sinus mucus recirculation. This can occur, as seen here in this video, when there's a scar band that forms after the maxillary sinus has been opened. And as the mucus flows up toward that natural os and then comes out of the sinus, instead of flowing toward the nasopharynx, it's going to come down around that scar band and travel back into the maxillary sinus, which will then carry it back up to the natural os, causing this looping of the mucus to occur. And this can, if there are bacteria or other inflammatory elements or infectious elements that are in that maxillary sinus, this constant looping can potentially lead to infection within the sinus. And this is just a video showing what that mucus recirculation looks like. You can see the scar band depicted nicely there between the natural os and the rest of the maxillary antrostomy. So there are a number of factors that can impair mucociliary clearance. So there can be primary ciliary dysfunction, such as seen with primary ciliary dyskinesia, where the motility of the cilia themselves are affected, which would reduce mucociliary clearance. And there's also can be acquired dysfunction of mucociliary clearance. So infection, like I mentioned previously, there could be virulence factors from common respiratory pathogens that can inhibit mucociliary clearance, either by reducing CBF, potentially affecting the viscosity of the ASL. In some situations, also potentially loss of cilia themselves. Another example where mucociliary clearance is impacted is in cystic fibrosis, where there's chloride transport dysfunction, which leads to buildup of thick mucus in the ASL and a decrease in ciliary beat frequency. So in conclusion, the sinuses and nasal airway have important physiologic roles as the first point of contact during respiration, and mucociliary clearance is an important part of the innate immune response. So some key points as well. Nasal airflow filters and humidifies inspired air. About 15% of airflow at physiologic levels passes through the olfactory cleft. Mucus is a gel layer with barrier infiltration and immunologic properties. And mucus and mucociliary clearance are an important part of the innate immune defense. The airway surface liquid depth and viscosity are determined in part by ion concentrations and gradients. And changes in ion transport can affect mucus viscosity and mucociliary clearance. And here are some suggested readings of 3 papers that can discuss some of these important physiologic properties and different roles for some of the cells in the upper airway. And here are the sources for this chapter.